Hi everyone. I know that I'm today maybe a few minutes earlier than normally here in the class, so I'm just gonna give a minute or so for the last people to join us before we make a start for today's material. In this time, if you have any questions, feel free to just shoot them out. I know that there was quite a lot that we covered on Monday in terms of the lecture material, but my encouragement is that try to stick to the big story, the big picture. Having said that as well, just to give you a heads up, I do think that today's class is probably going to be a little lighter, and I'm not even sure if we'll take the entire hour and 15 minutes. But while we're waiting for the last people to join us, if you have any questions, please don't hesitate to post them in the chat or unmute yourself and speak up. And of course, I'll be here around at the end as well. I usually get some excellent questions at the end of the class. So we'll start in just a minute or so. Well, I think I want to be appreciative and respectful of everyone's time. So we'll just make a start at this time, working through the material that I had for today. So what I was hoping to do today was to discuss with you about the lab that we have scheduled for today or for this week. And that lab really ties in very closely with everything that we discussed in the Monday's class. So on Monday, we discussed about two processes, mitosis and meiosis. And we're going to be focusing on mitosis on this lab. Normally, when we do it in person, it is a great chance for students to see that this really happens. You can really see this, all these diagrams that we were drawing and so on. You can really see it in real life. And I do realize that taking labs to an online setting is always challenging. So we've done the very best we can of making it involving, making it so that you actually get a chance to have at least the same skills and same knowledge as that you would have gained in an in-person class. I do also acknowledge that there are challenges, but I think that if you take the time, read through the lab and work through the lab, you will definitely be geared with the same information. So before we get too far, I think it's a good time for us to recap a little bit of the important concepts that we spoke on Monday's class. So one thing that we noticed, we had two types of cell division processes. First one of these was known as mitosis. And mitosis is really what applied to all cells with the exception of the sex cells, but all cells, and that was involved with the processes such as our growth, our tissue maintenance, repair of tissues, and so on and so on. So we see mitosis taking place all the time in pretty much all of the cells of a multicellular animal. The big thing with the mitosis was, as I'm sure you remember, that we started with 100% of the genetic material in the parent cell, and the resulting daughter cells contained also that 100% of the genetic material. And what's important at this point is to remember and recognize that every single cell in your body that has a nucleus, every single one of those cells contains instructions for everything in your body. It doesn't have to be a cell that's specialized for that very purpose. Let's talk about, for example, about the eye color or what way the hairline runs. Regardless whether it's a cell that's involved with the hair or involved with the color of the eye, every cell will contain that information. So that's why what we saw that any cell is required to contain that 100% of information. It contains the information for everything, all that you are in terms of a genetic standpoint. Things got a little bit different when we talked about meiosis. And we remember that we saw the process of meiosis only in production of two types of cells. So egg cells, oocytes, and spermatozoa, so sperm cells. Those terms that I'm using are the probably medically and biologically more correct terms. We do tend to be sometimes lazy and talk about eggs and sperms, but do not get thrown off if you see those proper terms being used as well. And what we remember, and I think that everyone at this level knows, that a new individual is a creation of both of its parents. So mother and father contribute both, if it's a biological child, 50% each for the genetic composition of that new offspring. So for that reason, we cannot have that each one would contribute 100% because we would end up with offspring that has 200% of the genetic material of a normal individual. And that would only get worse with every generation around. And what we end up finding really easily and really fast is that even slightest of amount of extra or slightest amount of too little chromosomes causes significant conditions for individuals. So we don't want to go anywhere else but to that 100%. So for that reason, each parent contributed 50%. That could be symbolized with N, and the offspring contained 100% of the genetic material, or 2N, if you wish. So meiosis is a little bit of an exception, but it is an important process, so that's why we spent some time talking about it. But on today's lab, we're going to focus on what's easily observable, what we can easily get material and data for, what we can easily study in a lab, and that's going to be the process of mitosis. And we're going to be using a model organism to study the process of mitosis. And I think I've mentioned the concept of model organism in the past, but I don't think we've gone into great depths about why model organisms are so popular in science. So when we're studying of things that are important when we're wanting to understand humans, there are some limitations what sort of experiments we can do with humans. These limitations might be because of ethical or legal reasons, but they can be also for many other reasons. For example, a human individual's lifetime is rather long for studying something such as how genes and genetics are passed from one generation to another. We would have to run studies that run for hundreds of years. So obviously that's not the best way to tackle studying genetics. So what we do, we find species that are very human-like in those characteristics that we're interested, and species that we have studied extensively, so we really understand these processes on those species. And usually these species have characteristics that make them also ideal for conducting research on. These can be, for example, quick turnaround time of generations, or they can be easy maintenance and management of these individuals. So they're not taking huge space in a lab, or they're easy to keep alive. They are things that are usually in ample supply, and also costs do play role at certain point when we're doing large scale research studies. So some of the most common model organisms we use in science include, for example, lab rats, fruit flies, but it can be also non-animals. So we can use, for example, many plants to study some very, very core and basic concepts that apply to humans. And we'll discuss in the next week's module, I believe, about Gregor Mendel, who conducted a lot of early research about human genetics by using actually pea plants as a model organism. So that's not to say that humans and peas are the same thing, but a lot of the same processes as we see with the human genetics, we can actually model with pea plants. And of course, pea plants have a quicker turnaround time from one generation to another. They're easier to manage. It's less ethically problematic to force certain characteristics on a pea plants to make sure that all offspring show a certain characteristic. That could be very, very problematic with human subjects. So we're not going to use peas actually on this lab, but we will use another well-available, quite common model organism. And that model organism is going to be an onion. And we're going to be looking at a specific part of an onion plant. We're going to be looking at the very tip of the root of an onion. So when you look at whether it's an onion that you would use for cooking, or if it's a spring onion, they all have these little roots at the very end of them, and that would be the root system of that plant. Typically these end up drying up and getting cut off or breaking off if we store these plants for significant time. But what I do, and what is commonly done in other labs as well with the students, is that we actually get onions and bring them back alive and bring their root system back alive. So after a while, the root system, if it's in right conditions, it will start growing again. And we're going to be interested only on the very tip of that root. And the reason why we're interested on only very tip of that root is that that's going to be the part that's actively growing. So it's growing length for the root. So to grow, there's going to be a lot of cell division taking place. And that should be reminding you what our chapter was all about. It was all about looking at the cell division process. So we can study the essential core concepts that relate to the human cell division by using onions' tips of their roots. And that's exactly what we're going to do. And for anyone who's wondering about the attendance, I have questions that I'm going to be using today. So let's take the attendance in just a few moments. So since each of these cells at the tip of an onion root is divided independently of all other cells around them, each of these cells is going to be at the different stage of the cell cycle. So by looking at single onion root's tip, we should be able to see quite a broad range of different cells that are at different stages of the cell cycle. And this includes the active divisions part and also the non-active division part of the cell's cell cycle. So this slide that I'm sharing here should be largely revision from what you might remember from what we discussed on Monday. So what we're seeing here is that we had separated 4 specific phases or stages of the cell division for the process of mitosis. Interphase was the stage in which cells spent most of its time. And we could see that when we looked at the previous diagram on that kind of orangey color of this cell cycle. So largest part of the cell cycle is going to be this interphase. And during the interphase, cell looks very much like what we are used to seeing when we talk about animal or plant cells. So we are used to talking that we see nucleus that stains darker when we use specific staining techniques to study microscopic cells. Well, once we move on from the interphase to this actual mitotic stage, that can be further divided into 4 different stages. So the first one was prophase, and we saw that the nucleus disappeared during prophase. We also saw 2 important structures to appear on the cell. We saw the centrosomes and we saw the cell spindle appearing. And both of these were going to be very important in subsequent stages of the mitosis. The second stage that we talked about is called metaphase. So during metaphase, what we saw that these chromosomes tended to line up on the equatorial plane. So they all arranged nicely on a one single line. And this is where the centrosomes and spindles start to play a role. The next stage after that was anaphase. And during anaphase, each of these chromosomes is split into 2. So we end up with just chromatids going to each end of the cell. Another thing that we saw that the cleavage started to form. And this is in case of animal cells. So this depression of where the cell will be pinched into 2. If it was a plant cell, and we will be using plant cells as a model in this lab, what we see is simply that a little bit of the cell starts to grow at both ends. And slowly the empty part, the gaps get filled in. So that was our anaphase. And the final stage that we saw was our teleophase, during which now we get back to resembling something that we saw at the very start. We get actually 2 cells instead of 1 cell, since it's been pinched in 2, and we have nucleus reappearing again. So I hope that that kind of rings bells and is very familiar to what we have talked about. And really the role of these labs is to take concepts that we have talked about and reinforce them, sometimes add a little bit more to the story. And one thing that I hope that you will take away from today's lab as we're reviewing it together is that even though we talk about these stages of mitosis, this is what you get told in textbook. In reality, life isn't quite as clear-cut clear, or as quite as clear as we see on this process that we have discussed. So let's have a look of that in a little bit. What I do want to do at this point is to take the attendance. And for taking the attendance, I have a question that really we haven't addressed on this chapter, and we haven't addressed even on our review. It's things that take and pull from the information that you have learned earlier on this course. Also a little bit going to your general knowledge. I'm asking you that based on everything that you know at this stage of biology, which of the following would be a useful tool for studying different stages of cell division in this lab where we're looking at onion root tip? And there's going to be one that's especially useful tool for studying these onion root tip cells. So if you don't mind, I would ask you to post your answer to the chat. That's a way for me to get the attendance. The keyword that you can see there is that we're studying cells. Which one of these could be a useful tool for that? And everyone who answered that a microscope would be useful for studying a cell is definitely onto something. So really you see that that's why we started early on this course by going through basics, how to operate microscopes in an efficient, safe, proper manner, because that gives us a foundation that we can then use that to work through other things in this course. So even though you're not in person in a lab, we have learned how to use microscope properly, and now we get to see what that microscope and using it properly could be useful for. So I'm just gonna take a second here and look. I think it was mainly answers to the question that came through, but because every now and then there's a question that I miss on the chat, I'm just jumping there, but I don't see that there would be anything. If you have questions, do remember that you can always ask them. So feel free to unmute yourself, speak up. If you post them to the chat, I do try to monitor the chat, but sometimes I miss if something comes there, so don't feel that I'm ignoring you. Now that we made a good start of this lab, the next thing that I want to talk about with you is how would we prepare the actual sample that we're going to be studying on this lab. So let's talk a little bit, and this is really what we would be doing in a face-to-face lab. I do realize that online experience is not the same, but due to the global pandemic, we moved quickly online. We are currently working hard to develop similar experience for you in a virtual setting. And one of the things that we're very much looking is looking of lab kits that we could probably send to our students. So how we would prepare this sample from this onion root tip that I said that I usually get for my students. I start growing some onions before a class. We want to take only a very, very small part of that root tip. We want to cut only the very tip part because we don't want to see the cells that are just fine and hanging out there. We want to see the cells that are dividing. We might have to use, since we are talking about plant cells, and plant cells do have a hard cellulose wall, we want to make that cellulose wall a little bit easier to break, a little bit easier to penetrate. So we might use a few different chemical tricks to do that. Usually I boil these tips of or we boil them in a glass or put them in a warm water bath with some chemicals that make the cellulose wall become a little bit softer. At that point, we do want to make sure that we do only study a very thin section of the root tip. Remember, light microscopes relied on the light traveling through the sample. So if we have too thick of a sample, first of all, the light is not going to travel through. And also we end up with a bundle of cells on top of each other. So we only need a very, very thin section from there. One thing that you'll notice when you end up doing more microscopy, especially if you end up taking classes such as microbiome or studying microscopic samples in general, is that we often use different kinds of stains to make different structures of the cell really stand out and become more obvious. The stain that we use in this case is ideal for staining the chromosomes, especially dark. So it's really a field of art, and I had a pleasure of spending quite a few years at the university specializing on which stain to use on which cause, what are we trying to accomplish, and it really depends on what do you want to see, what kind of a staining process you utilize. And again, we're kind of taking a little bit of that information that we talked earlier on in this course about good practices of using microscopes. We might want to have this cover slip on top of the sample as well. We actually use this in this particular lab to also break up the tissue organization a little bit, so we get loose individual cells to the slide, so we can study them. So that's how we would prepare a sample to study from the tip of an onion root, so we can study that microscopic sample. And that brings us to a really nice, simple, straightforward quiz question that based on this, how would you prepare a microscope slide of an onion root tip? And there might be more than just one correct answer. If you didn't get a chance to post an answer for the attendance question, this is a good chance to get that attendance. If you did already get a chance to post an answer to the attendance question, that doesn't prevent you from taking part. What I do after every class that we have, I do actually take a record how actively you've been involved, and those will count as extra credit towards the end of the course. So being actively involved in a class definitely counts as a positive thing. It's not required, but I do want to acknowledge if someone takes part in a class. So if you do want to, you can give it a go. I will show the answer in a second. How would we prepare a microscope slide sample of an onion root tip? And really, the correct answers are the first three answers. We saw all those as the steps that we took earlier on. And of course, we do not need to purchase pre-made slides. We are completely capable of making those ourselves. Sometimes we do use pre-made slides. We purchase those, or you might have lab techs preparing those for you, simply because we might need to use sources of tissues or cells that are harder to come by. So good job for anyone who picked up all of those three correct answers. So what you would typically see on a microscope once you start looking for this onion root tip specimen would be something like what you see here. So we actually see the shape of the onion root here, and we see that the very tip part here contains most of the cell division process. So the cells are dividing at the very tip of this onion root at the highest rate. So we have the best chance of finding what we want to see, which is different stages of mitosis, by focusing on these particular cells at this part of the onion root tip. And what we end up looking is kind of these individual cells. They might look like little boxes or old school TV screens. And often they are arranged in kind of a row-like manner. So you'll notice that plants tend to have that sort of a preference. And what we are focusing within of these cells, once we've identified individual cells, is to look for individual chromosomes. These chromosomes would stain dark with the stains that we use. Here I have a little bit higher magnification picture of what one might see. And you'll notice that there's a lot of cells where we can simply see the nucleus, and that's about it. But every now and then you can see a cell that's either about to or just undergoing a cell division process. And you can really see either chromosomes having condensed or chromosomes being organized in a particular manner that aligns with the stages of mitosis that we have learned about. If you're looking at this presentation on a small screen, I realize that it might be a little hard to see, so I do have better pictures coming up, but already at this early stage, you see here and there, we have evidence of different stages of mitosis taking place. And again, this is the reason why we learned how to use microscope, how to get into those higher magnifications, and how to safely do that, not damage the samples, not damage the microscope. So let's have a look quickly, a reminder of what we just discussed about the stages of mitosis. And the one stage that I added was our interphase. And then the mitosis proper included our prophase, metaphase, anaphase, and our telophase. And each one of these were characterized by the chromosomes either being arranged or appearing in a certain way. So if we just study the chromosomes and we see any of these characteristics that we're seeing here, we would be able to conclude at what stage of the mitosis the particular cell is in. And of course, that can be also either beginning or of ending of the cell division process. So we're going to be going through these different stages of mitosis, and we're going to be going through them by looking cell by cell what's going on. And here I've actually kind of brought a different view from what we have already seen of this microscopic view of a onion root tip, and you can see I'm kind of highlighting or circling a couple of cells where we can see something very interesting happening. We no longer see just the nucleus. We're actually starting to see the chromosomes condensing, taking the shape of the chromosome. The DNA is taking the shape of the chromosome, and we can see 2 different stages of the mitosis already at this view. So this brings us to this view that I have kind of prepared for you. I've taken a couple of different methods of visualizing and staining cells at their different stages of mitosis, of mitotic division. And you can see that our focus is on these darkly stained parts and identifying which stage each one of these might represent. So the first picture that we see is going to be our interphase. Still in the interphase, we do have the nuclear envelope present. The nucleus has stained dark. Once we move to the prophase, one of the things that we start to see is that the nuclear envelope has now disappeared. That's no longer present there. And we can see that the genetic material has taken the shape of chromosomes, but these chromosomes are all over the place. They are not organized in any particular clear manner. When we get to the metaphase, in metaphase, we are seeing that these chromosomes are lining up on an equatorial plane. So we remember that was one of the important characteristics of the metaphase. Unfortunately, with a microscope on this level, using light microscopes that we have available in labs, we are not going to be able to see the spindle or other finer structures. But we do see the chromosomes, and they're enough for us to make a determination of what stage we're looking. The 4th stage is going to be our anaphase. And you remember during anaphase, each chromosome was pulled apart into 2 chromatids. So we're seeing that happening there. And finally, telophase was our last stage. And in that stage, we now have 2 cells. And each one of these cells now have the nucleus that has reappeared, the nuclear envelope is present, and we can tell that stage from there. So now we're starting to see that everything that I've told you so far on diagrams actually exists. And even with just basic tools that we have in our basic biology lab, so basic microscope, we can start to see those. Of course, the diagrams that I show you are often a little bit more polished. They are a little bit more simplified. They might not show every single chromosome at the same time, but we really do see the same stuff when we look at the real life. And this brings me to a nice reminder again of this summary of mitosis. And I think we've gone through it a few times now, so I'm just simply asking you to notice how the very same things that we have discussed we can see on these actual microscopic images of the cells. And based on that, let's just check that everyone's with me at this point. I have there 5 different stages. And I'm asking you, based on those photographs, there's 2 pictures of each, to identify which one of these would best represent interphase of the cell cycle. Remember, the interphase was the stage in which the cell spent most of its lifetime. So again, a good chance to get those extra credit points towards your final score. And if you did answer A, you were absolutely correct. And we just went through that together. So I hope that that wasn't too much of a curveball and that kind of felt like an easy one, but helped you to learn to really look at the samples. Also, I just want to make sure that everyone's with me, and I'm asking you to pick from these that which list of different stages would be correct to describe the different stages of mitosis in a correct order. I have left the interphase away. We're looking at the mitosis proper, and there should be 4 stages. The 5th option that I've listed there as an option E is that none of the options that are provided is correct. And if you did answer A, you are very correct. So clearly you have this process of mitosis worked out great, and you've got this. So now that we've kind of done all the groundwork, we really should get into actually looking of the results that we're gonna get in this lab. It is a nice thing to learn how to prepare a sample. It is a nice thing to learn how to analyze a sample, but we're doing all this for a very particular reason. We're doing it to learn some new information. And what we're wanting to learn is that how much time each of these, how much time of its life a cell spends in each of these stages. And if we think that we had an onion root tip where cells were dividing totally at random, independent of each other, well, we're expecting to see cells from all of these stages. And I'm just trying to get a pencil to work again. So we're expecting to see cells from all of these phases. And based on how many cells we see on each of these stages, we can then work out that, well, cells at large are likely to spend most time in this phase and least amount of time in this phase. So simply by starting to count cells and starting to classify them into one of these 5 different phases, we can start to make sense that, well, how long does the mitosis proper really take? And normally, if we were to do this in a lab, I would recommend that one student looks into the microscope and another one keeps a record, and you just start going through these cells on these kind of a row-like arrangement of what you see on a microscope, and you just call out what stage each of these cells is at. And we would do this in each group, but then we would also combine the results from each group together to get a class data. And last time I did this, I counted 115 cells. And out of these 115 cells, 89 were in the interphase. And we can actually see that nicely on this picture that we're seeing. Most of the cells do have the nuclear envelope. Nucleus is clearly stained. We do not see individual chromosomes. We do see some cells, though, that are going through some of these mitosis proper stages. So when I counted these, I got 5 in prophase, 6 in metaphase, 6 in anaphase, and 9 in teleophase. phase. But all this data is telling us a very clear story of how long of a time a cell spends in each of these stages. So let's take this as a chance to explore that. But I do want to make a disclaimer at this point, and this is going to be something that's very, very important to remember. Although I try always my best to make your life as easy as possible, so I give you these diagrams, I talk about phases. In reality, if you were to do this lab, one thing that you would find out that it would be hard sometimes to tell apart. For example, is this cell now in prophase or is it in metaphase? Are the chromosomes present there, but are they kind of all over the cell, or are they lined up nicely on an equatorial plane? So it might be blurry, if you wish to describe it that way, to tell apart that in which phase really the cell is. And this is because cells don't just jump from one phase to another. They go through a continuous process. So rather than being individual stops, it's kind of a continuum that we're seeing. So we're slowly moving through these phases, and there might be cells that are in between two phases. So it becomes harder in real life, but we can still do classification, and we just do our best. But once I did this classification, I got my data, and I've included that data here, as you could see. And I actually have 4 statements here, and one of them is incorrect. So 3 of the statements are correct, and one of them is incorrect. And I'm asking you on this quiz question to spot the one that's incorrect. So which of these would not be correct? A, cells spend majority of their life in the interphase. And we saw that we had most cells in the interphase. So that statement seems to make sense. The second question that I have is that would it be that 7.8% of the cells observed were in telophase? Well, if you get a percentage, how many percentages? 9 is out of 115. I think it's somewhere close to 7.8 percentages. So we would be inclined to agree with that as well. Well, the next statement that I have proposed there is that the number of the cells counted in each stage, so all of these add up with the total number of the cells counted. And if I do remember that I did this correctly, mistakes are always possible, but I do think that they add up to 115. So we are left with only one statement. And that the statement would be that we could not draw any conclusions based on this lab. And I would hate if that was the case. I hope that we are able to draw some good conclusions from this lab. So that leads us to notice that answer D was the incorrect answer. And having said that, now we get to the very last question on the lab, very last question on that you have to answer when you're working through the lab, and also very last question that I have prepared for our review together. I am asking which of the following is true regarding the stages of mitosis. And there is one statement that's correct and 3 statements that are incorrect. So I'm looking for that true statement. Is it that the mitosis is a continuous process, not a series of separate events? That would be A. Is it that all of the cells within the same area progress through mitosis at the same phase? That would be B. Would it be that most of the onions have been genetically modified, which has disturbed their normal cell division process? That answer would be C. And answer option D would be that biology is way too hard and complicated, so we should not study it at college. And I'm going to be honest, if you do pick the number D, I am going to be very saddened, and I might be emailing to you, and we might be trying to figure out what can I do to help you. So it definitely is not going to be answer D. The correct answer, of course, was option A. Mitosis is a continuous process, not this series of separate events, although so far I've been teaching you that it would be these separate events, but in reality we move through those. So it's a continuum rather than a set of stages. I hope that that made sense, and that really brings us to the end of everything that I had planned for this lab for us to discuss together. Like I said, I do realize that we went through a huge amount of information on our Monday class, and I do realize that I also kept you a little bit of an overtime. So this is really all that I have planned for today. I will be here in case you have any questions about this lab, or in case you have any questions about anything of the chapter material that we covered on Monday. So I'll be around to answer any questions, tackle any challenges. But if you got everything you need out of this review of a lab, you are done for today. I really appreciate you joining me. And like I said, I will take the attendance based on your answers, and I will also make sure to record your extra credit. I know I haven't published that yet on the gradebook, but as soon as I get a chance, it has been a busy semester for everyone. And as soon as I get a chance, I will be definitely posting your extra credits. Fear not, even if you're not seeing it yet, you will be recognized for your work and for being actively involved in the classes. So on that note, that's all I have for today. I hope you have a very good rest of the week. And as always, I'm here to help. I know I have few messages on Blackboard that I haven't had a chance to answer yet, and I will do it as soon as possible, but I'll also be here to answer. Have a good night. Thank you. Thank you so much. I appreciate all your hard work in this class. Good night. Thank you so much.
Hi, Professor.
Yes, can I help with anything?
I have a question.
Let's see if I can help you.
Do you have you seen my message that I sent on Tuesday about my homework?
Have I answered to that yet?
No,
Probably
you
not.
haven't.
If I haven't answered yet, I probably have missed it. Would you like me to stop recording so we can talk off record? Is that something that would be nicer?
It doesn't matter. I was just wondering about my homework. I missed one question on it, which was the statements for mitosis and meiosis.
Okay.
And I was looking through it and I was trying to see which ones I got wrong, but to me they all look right, and I don't know if I'm missing something, but for any chance, can you check if what I'm missing?
I'm
Or I
gonna
could
be
just...
honest that probably you have not missed anything. I sometimes make mistakes too, so it might be that I just have something wrong. So give me until tomorrow, and tomorrow you should receive a message from me. And if it has been an error in a way that I've set up the question, you will definitely get those points, and I will fix it for everyone. I really appreciate you bringing it to my attention, because like I said, we do this and sometimes we as well work at weird hours and mistakes happen. So I will definitely check on that. If it is just an error at my end, I will let you know. If it is something that you missed something, I will let you know exactly what you missed. So thank you for sending me that. I really do appreciate it, and I will get back to you by the end of tomorrow. If you haven't heard back by the end of tomorrow, please don't hesitate to drop me a reminder message. I normally try to get back to, but sometimes things slide through the cracks, and I don't want you to be waiting if I've totally missed your question.
Thank you so much. I'll be
Thank
looking
you.
forward to
I
it. Have a
appreciate
great night.
you.
Thank
Good
you.
night.
Good night.
Hey, Professor.
Yes, can I help with anything?
No, I just wanted to be able to tell you that I'm sorry I missed on Monday. I had a lot of things coming up in the past couple of weeks, so I was just letting you know that I still got plenty of time to do all the assignments and relook over the recordings and stuff. I just wanted to
Absolutely. I
apologize
so
for that.
appreciate you letting me know. Please don't feel bad about that at all. I totally get it that life happens. And as long as you're able to make one attendance a week, you're doing excellent. So you should be able to get all the information from the learning module. I think I have very extensive videos there, or you can watch the recording. It's either way. There is a lot of material this week, but don't get bogged down with the little details. Get the big story. And I think that we covered pretty much the most important things already on today's session. So you should be absolutely fine. And knowing your standard of work and knowing how hard you work, I think you'll be just fine.
All right, thank you. I just want to let you know that.
Thank you. I'll make sure that you get an excused absence for that day.
All right. You have a good
Thank
one.
you. Have a good night. Bye.
Thank you. Bye.
Professor, I was wondering if that was it for like the session. Like we did
That's it for
attendance
this session.
already.
Yeah, that's it. That's all I have for today.
Oh, okay, because I guess I kind of missed it. I was like, oh, it's like that's it. So,
No, I know that it was a little shorter today, and I'm still here writing things down. But that's it for today. That I have nothing else to share. If you missed any part, there's a recording that's gonna be posted in about half an hour or so of this session. But that's it. The lab itself is, I think, 6 questions long, so there's
okay.
not that many questions.
Do I just put like my name down since like for attendance or?
Yeah, just type your name to the chat and I'll have it. I do take notes, but I'll make sure that you get attendance. If you just type your name to the chat, you're all set, and that's it for the night.
All right, thank you so much, and I hope you have a good night.
Thank you. Thank you for checking.
All right, bye-bye.
Bye. And that really completes the recording for today. I appreciate everyone who joined me either in person or through a recording, and I hope you have a very good rest of the week. I'll see you next week again. Thank you. Bye.